In this unit, we will discuss the organization of the cell, from the role and structure of the cell membrane to the function of the eukaryotic cellular compartments called organelles. In this video, we will focus on the lipids and proteins that make up the structure of the cell membrane. All cells are defined by the presence of a cell membrane. The role of the cell membrane is to regulate what molecules can enter and exit a cell, essentially keeping the insides in and the outsides out. As discussed in Chapter 2, the major type of lipid found in the cell membrane are phospholipids, amphipathic molecules which are able to associate together into a lipid bilayer. Neighboring phospholipids associate with one another by van der Waals forces between their fatty acid tails. Additionally, individual fatty acid chains are able to flex and bend, thus the membrane is fluid and flexible. Phospholipids are able to move horizontally in the plane of one layer of the lipid bilayer, but they are rarely able to flip from one layer to the other. The amount of fluidity in the membrane depends on the strength of the interactions between the fatty acid tails. In Chapter 2, we discussed the difference between saturated and unsaturated fatty acid tails. The more saturated and the longer the fatty acid tails, the more tightly they are able to pack together, decreasing the fluidity of the membrane. So this membrane here would be less fluid because of the increase in saturated bonds, whereas this membrane would be more fluid because of the increase in unsaturated fatty acids. Cholesterol is an important component of animal cell membranes. While it often gets a bad rap because of the association of high cholesterol levels and heart disease, your cells require a certain amount of cholesterol to exist. It is important to note that cholesterol is only found in animal cell membranes because it helps support the membrane by maintaining fluidity as the temperature changes. At normal cell temperatures, the interaction of the rigid structure of cholesterol with the phospholipid fatty acid tails reduces the mobility of the phospholipids, reducing their fluidity. But at lower temperatures, cholesterol prevents the phospholipids from packing tightly together, increasing membrane fluidity so that it does not become too rigid. This is not required in plant cells because the presence of the cell wall provides support for the cell membrane. The cell membrane is not only made of lipids, but also contains numerous proteins. While the function of the phospholipids is to produce a semi-permeable barrier to control what can enter and exit the cell, the function of the membrane proteins is very diverse. These proteins can serve as transporters, allowing molecules to cross into and out of the cells. Some function as receptors, which allow the cell to receive signals from the environment. Enzymes are able to catalyze chemical reactions, or anchor proteins, which attach to other proteins of the extracellular matrix or other cells to maintain cell shape and structure. Membrane proteins are classified into two types depending on how they are associated with the membrane: integral membrane proteins or peripheral membrane proteins. Integral membrane proteins are permanently associated with cell membranes and cannot be separated from the membrane without destroying it, while peripheral membrane proteins are temporarily associated with the cell membrane through weak, non-covalent interactions. As mentioned earlier, the phospholipid bilayer is a flexible, fluid structure. This means that the proteins embedded in it are able to move around the cell membrane similar to ships moving on an ocean. The fluid nature of the membrane is able to be observed using a technique called FRAP, or fluorescence recovery after photobleaching. Watch this short animation for an explanation of FRAP and to see it used on a living cell.